All right. Hi, everyone. I'm Sean, the CEO of BioTuring. Welcome, everyone, to the BioTuring Single Cell Seminar. So, today we have a special guest, Ilya Korsunsky, the author of Harmony. And as everyone knows, single cells data are generating at exponential speed and create a lot of opportunity for exploring biology. But at the same time, it creates a huge challenge for computational community. One of these challenges is batch effect removal. That is, removing the differences generated, not from biology, but from different technologies. And at BioTuring, we process a lot of data, and we test a lot of tools and algorithms out there for batch effect removal. And there's several metrics there that we have to consider. One is the computational runtime, because not only the number of data sets are increasing, but the number of cells in each data set also increase so fast. The second is the ability to handle large data set. And the most important is the batch effect correction efficiency while preserving cell type purity. And of course, if we consider the last factor, there's still few tools left, including Leisure, including Seurat, including Harmony. But if you have limited resources, computing resources, and even without the computational resource constraint, I believe that the first tool that you have to try is Harmony. And this is my great pleasure today for introducing Ilya Korsunsky. Ilya is currently a PI at Brigham and Women's Hospital and Harvard Medical School. Before that, he did his PhD in computational biology at New York University. And let me give the time and space and stage for Ilya Korsunsky. Okay, Ilya, welcome.
Thank you, Sean. Yeah, thank you so much for the invitation and as well as the great introduction. I think you've really contextualized this this problem very well, and we started to analyze data. It sounds like around when BioTuring was being formed, we started analyzing these single cell data, and we were running into. We lived in blissful ignorance of batch effects, because in the early days we only had two batches that we were working with and analyzing for like 6 months, and then as soon as we got our third batch, then we realized, oh, this is going to be a big problem, and so we got to working quickly on some kind of integration methods, and trying to understand this problem very well. Anyway, yeah. So yeah. Without further ado, let me jump into the meat of the talk. And it sounds like it looks like there is going to be a chat box option, right? If there are any questions along the way. So definitely happy to field questions, any questions that you have along the way. But also hopefully, we leave enough time towards the end of the seminar to get to any questions.
Do you want the audience to ask the question at the time that you speak, or you want to leave the questions at the end, Ilya?
Yeah, I'm okay with during the talk.
Okay.
We can field the questions during the talk.
Yeah.
Yeah, there is.
All right, I can help you coordinating it.
Thank you. Yeah, yeah, if you could look at the chat box, I'll focus on the slides, and
All right.
feel free to interrupt me as if we were live.
Yeah, this is usually the style of a computer scientist and a mathematician.
For sure. Yeah.
They like to
Very
engage
happy.
and ask tough questions.
Yeah. Yeah. So please do. Please do. Okay. Can you see my slides right now in
Yes, I can see your slide.
presentation mode? Okay, good. Okay, great. So title of today's talk is the title of the manuscript. I added 2 words to it, theory and practice. This is where I really like to communicate with the community to make a leap between the math of behind the model, which I think is relatively simple, and how that connects to data analysis and practice. So as Sean said, I'm a PI. I started my lab this last July 2021, Brigham Hospital, Harvard Medical School. I'm affiliated with the Center for Data Sciences here, and my lab is housed between the divisions of rheumatology and divisions of genetics. And I'd like to start off the bat by thanking everybody who contributed immensely to this project in particular. I started and published this paper while I was a postdoc with Shomari Chowdhury's lab. Yeah, and then other people, including Jean Poru, Kevin, and Michael from other groups, provided very important feedback during the development process. So this is it. This is the paper. It was published in 2019, and it's available in R. It's available in. Oh, sorry, I skipped ahead. For full disclosures of funding sources, my lab is funded right now partly by NIAM's training grant, partly by Biogen, and I personally receive consulting fees from Mestech Therapeutics. Okay, so now the paper. So this is the paper. It was published in 2019 in Nature Methods. There's many implementations and versions of it out now. The 2 primary implementations are the one that I coded on the left here in R, and then the contribution of Kamil Slowikowski here was to code it up in Python. And there's also PyTorch versions on Python if you want to use Harmony on super big data sets. Okay, so this is an outline of where I want to communicate today. First, I want to talk about this problem because I think it's a tricky problem, and I want to motivate why we want to use it and what we want to get out of this integration of single cell data sets. Next, I want to go explain just some parts of the math and theory behind the Harmony model, and hopefully understanding a little bit of that math and theory will help you when you actually use Harmony in practice in your data integration use cases. And then once we go through these parameters, and then it'll motivate your use of certain parameter choices as you run Harmony. And then once we go through that overview, I'll go through 3 cases from standard to some non-standard uses of Harmony and integration. Okay, so let's start here. Let's talk about this problem a little bit. What is single cell data integration? I would say that there's roughly 3 reasons that you would want to use this data set integration. One, as Sean already said, is batch effects, right? If I profiled basically the same system from very similar donors, but I did it with 2 different technologies, or I did it on 2 different days, or I did it with 2 different technicians, right? And when I plot all of my data into some low-dimensional plot here, and the data completely separates by batch rather than by cell type, that's not great for my downstream analyses because I want the cells to cluster by cell type, not by batch variant. So clearly, this is a case where we would want to remove the effect of confounding batch. The other one, which we use a lot also, is real true biological effects. So this is actually a great example from one of the first integration papers from Rolls Atijah's lab, where he demonstrated that if we analyze PBMCs stimulated with interferon beta and non-stimulated, and then look at them all together, there's a very big effect that's introduced by interferon beta stimulation. But and so we wouldn't call this a batch effect necessarily, but it does impede our ability to identify shared cell types between the two, right? Because at the end of the day, what we really want to say is, what's the difference? What's the response of interferon beta in T cells? What's the effect of interferon beta in B cells, right? And so to do that, first we need to find the T cells and we need to find the B cells. So for that step, we want to integrate out the real biological effects. Then after clustering, go back and do some differential expression and ask, how does this biological effect affect different cell types differently? Finally, a third level of integration, which is becoming more and more popular now because the data sets are becoming more available, is that we'd want to integrate across different modalities. And we've done this as well. We do this actually in the Harmony paper. We integrate single cell from 10x with a MERFISH data set. So this is in situ spatial transcriptomics. And then this is an example from another paper from Rahul's lab where they do a very similar kind of integration from single cell and in situ spatial transcriptomics. And the idea here is that each data set has something the other one doesn't. And by integrating the two on some kind of shared feature space, we get to make use of all the data that's present in one technology and present in the other technology. And so this is also something I would not call batch effect, but a very useful use case of Harmony and single cell integration in general. Okay. So hopefully now you're motivated, or maybe you recognize some of the use cases that you're interested in in those three levels. So now let's walk through the ideas behind Harmony and then the math behind Harmony. So overall, with single cell integration, the goal is to remove some kind of effect that you where some batch effect, but keep the cell types, keep the cell type effect. You don't want to remove that. And so in a very simplistic kind of cartoon, if we start with this unharmonized data where cells group both by cell type, here's shape, and by data set, which here is denoted by color, we want to remove the effect of color, but keep the effect of cell type shape. The first thing we started with is just a simple naive linear regression. So you can imagine that this is a nice place to start. And it worked reasonably well in bulk RNA-seq data and in microarrays before that. And so it was a reasonable place to start here as well. So let's see kind of picture geometrically what happens if we apply linear regression to this toy data set. So linear regression geometrically basically says, compute the center of each data set, of each color, and then match the centers or move the centers so that the centers match, right? And so let's see what happens when we move all the cells to make their centers match. A few things happen. First of all, the colors do not mix well. And we actually get mixing. We used to have some things that were well mixed, so like the green and the purple squares, and now they're not well mixed at all. And we have the danger of moving these star-shaped and fish-shaped cells closer together. So we've done everything wrong with global linear regression. We've not removed the effect of color, and we've moved different cell types closer together, which is not good. And this doesn't work. This is not unexpected. This is something that was expected and called out from some of the early batch correction algorithms. So this is another one of the very early batch correction algorithms that are correct. In their discussion, they basically say that linear models don't work because the batch effect is cell type dependent. You can't apply the same correction factor to every cell type and expect it to work. So we turned that, they use that as a major limitation of why you cannot use linear regression. We use that as a challenge to say, okay, let's develop an algorithm that uses linear regression that learns cell type specific batch effects. And so this is what became Harmony. So this is the idea, is that let's actually move into this part. So this is what Harmony does. Harmony, instead of applying one global batch correction factor, first it's going to try to break up the data set into different regions. So here it breaks it up into 4 regions. And within each of the regions, we're going to compute a batch effect centroid, and then move those and then figure out what we need to do to those centroids in order to move them into the same space. So it is still linear regression, but instead of doing one global linear regression, in this toy example, we're doing 4 linear regression problems. And notice that we specifically made this data set to have several types of manifold structures that we expect to see in single cell. So some cells will be clustered into discrete blobs, right? So this is like cluster 1 here, right? They're pretty round. Clusters 3 and 4 represent what we might call a trajectory, right? So here, and it's very important that when we integrate the data, that we don't turn the trajectory into discrete clusters, or vice versa, right? And so the way that we do that here is that each cell gets an average correction factor. So the cells that are really on the boundary between cluster 3 and 4 are going to get an average of these 2 arrows that are the correction factors for cluster 3 and 4. And so while cluster 3 says move yellow cells down and right, cluster 4 says move yellow cells down and left, the yellow cells on the boundary between 3 and 4 are just going to move down and not left or right. And so by doing this soft clustering and soft correction, we are able to maintain trajectories and integrate trajectories well, even though we are doing an underlying clustering under the hood. Okay. Oh, so it looks like we have our first question. The question is there a way, besides plotting, to assess how successful my Harmony integration was? Will it affect the differentially expressed genes or improve my results? It's a very good question. Very broad question. I think I would like to maybe save this one to the end because it is such a general question. I'd like to answer it in the context of the use cases that I will present after. So let's save this one to the end. This is a great question. Okay. Other questions. Does Harmony take batch classifiers into account into this calculation? I'm not sure what, oh, okay, just to clarify. Oh wait, so Johnny, can you clarify what you mean by batch classifiers? Okay, so to regress out data set in this example, right? So that's what we're doing. We're regressing out data set. That's right. That is what we're doing in this model. Yeah.
I see. So just to clarify, you would be inputting that as a as like something in the function for Harmony. You would specify like regress out data set as a batch.
Exactly. Yeah. So we can regress out multiple factors, but you do have to label the factors, right? You do have to know what you were trying to model and remove.
Yeah. And do you ever see any situations where you have like a spike in control, kind of like a shared across? Because we've been thinking about doing some study designs like that, but I don't know if Harmony's built to do that.
Spike in controls. So how would you use it in that case? Or what's the?
I would use like a 293T spike into every batch of experiments, and then because we know that those aren't changing. Because if you do experiments across time or like chemicals, it changes the biological signal, so it's not like a straight.
Yeah. So I think there's batch effects that come from lots of questions. Okay. So I'm going to try to keep my answers brief, and anything I can't answer in 30 seconds, let's let's take a discussion offline, because
Yes.
these are all very interesting. So I think for spike-ins, you can definitely imagine a case where you can do it, and harmony is just regression, and so we can definitely, I don't know, figure out how to kind of include that information into the regression model, right? What I would be careful about is assuming, again, that if I have a spike in of one cell type, right, in my batch effect, let's say over time, right, and then my actual cells of interest are not that cell type, or not 293, right? Batch effect is going to affect your positive controls differently from your actual cells that you're interested in analyzing, right? So it's not that batch is going to affect your spike in the same exact way that it's going to affect your other cells, right? So I don't think it entirely solves the problem. Yeah.
Yes.
Okay.
Yes.
So that's
Ilya, I want to propose how long would your topic would be? It be in 30 minutes or an hour?
pretty quick. Oh no, more closer to half an hour. Yeah. So that's why I'm happy to answer questions.
Okay.
So,
Okay.
yeah,
Because I just thought it can take into 3 hours talk easily.
yeah. Yeah. Okay, so Amrit asks, just to clarify, harmony corrects for local batches instead of global correction. Is it possible to do both? Yeah, actually, it's a very good question. It's something I want to include in the next model. Again, it is just regression, right? So we want to include, we can include a global intercept and an intercept for every cluster, basically, and that's definitely what I want to do. I think in practice, my gut feeling is that it's not going to change much, but it is more elegant to do it that way. Bill asks, what are the assumptions regarding a low-dimensional embedding? Does it have to be linear? Can it be approximate? Yeah, I mean, PCA is what we've been mostly using harmony on, and PCA does have a linear assumption. But when you actually look at the data, right, the PCs are obviously not well-behaved Gaussian distributions. There are outliers. There are multimodal distributions in the PCs, right? And so I don't know exactly how much we can break these assumptions before harmony fails. It's a very good question, though. But I guess intuitively, things need to be locally linear for it to work, right? So global linearity is absolutely not a constraint. But as long as you can divide the space into things that are approximately locally linear, then you're good. Cool. So that was Bill, Johnny. Okay. Wigwo asks, cell subtypes usually very close to one another. That's when it's difficult, for sure. What is the impact of harmony procedure on subtype distribution or classification later? How do you reduce potential artifacts for subpopulation? Okay, yeah, that's tricky, right? So trajectories are a great example, right, where things don't cluster into really neatly separable groups. So there's definitely some kind of underdeterminedness of the problem, right? That if two clusters are very close to each other, do I want to move them closer together because I think they're batchy, or are these two parts of a trajectory, beginning middle in one batch and middle late trajectory in the second batch? And I don't really, you can't really know that 100%, right? And so harmony kind of implicitly makes some kind of decision in the regression to say, if you're pretty close to each other, we're gonna integrate you. If you're pretty far from each other, we're not going to integrate. So it's definitely, I would never apply this blindly. I would always go back and ask, where do the genes lie after integration? Am I happy with this? Do I want to be less aggressive? Do I want to be more aggressive? We definitely analyze a lot of things, subtypes. We use harmony to, build a T-cell atlas with 60 cell types. We use harmony to build a fibroblast atlas where also everything looks like one big blob in UMAP space, right? So it's definitely something that's been useful for us, but I wouldn't apply it blindly. Applying it with NMF, this is actually interestingly something we're doing actively this is something we're doing actively right now, is applying harmony to NMF. And I'll say, watch Twitter or something, and we'll hopefully give you updates. In theory, there's nothing stopping you from doing that. You'll get some negative things after the correction, but otherwise, in theory, there's nothing stopping you from doing it in NMF. In your example, each of the clusters contains multiple batches. Can you clarify if the batches are not intermixed? If they're not intermixed and there's one cell type that's really just only belongs to one batch, nothing happens to it at all.
Yeah, there's a question from Eric. How does harmony decide? Sorry, from an anonymous.
Yes.
How to decide how many clusters?
Yeah, that's a very good question. There's a certain range. Yeah, maybe one or two more questions, and we can move on. There's a certain range of cluster numbers where you get very stable results, right? I'll give you an example of extremes. I think this is where it's very useful to know. So if I have one cluster, right, then harmony just reduces to basic global linear regression. If it's one cluster, if I have 1,000 clusters and 1,000 cells, well, I'm not going to correct anything because every cell is going to belong to one cluster, right? More or less. It's actually not entirely true the way we implemented harmony. If you give 1,000 clusters, it will kind of be robust to that because harmony right now makes it so that you can have redundant clusters. So if there's 12 real clusters and you give 15, you can actually look at it. 3 of those clusters are going to be collapsed in, folded in, and completely redundant with 3 other clusters. So there is some kind of redundancy and fail safes with this.
Right.
So I think it's a good idea. Yeah, let's move on temporarily until the next section. But yeah, let's keep writing the questions and we'll address them as they come in. Okay, so the basic way that harmony works is right there, well understood. The secret sauce sort of to harmony was, what if we get all these clusters and these clusters are only one data set in each one, right? Then you're not doing any correction at all, right? No correction will be performed in these cases. And so this is a thought experiment that we did, right? How to solve this, right? So let's say we have the situation where we have B cells, T cells from me and from Sean, right? If we just apply regular k-means regression here, because of the way that I drew the cells, regular k-means will group my T cells with my B cells and Sean's T cells with his B cells. And so that's not what we want. That's bad. That's one, it's wrong because it's wrong biology, and it doesn't give us the opportunity to compare my T cells to Sean's T cells, right? And so what we want is to apply a penalty that says clusters with only one batch are not preferred. We'd prefer to have clusters with multiple batches, right? And so that's what we did. We used KL divergence and chi-squared contingency tables basically to modify the objective function of k-means and Gaussian mixture models to be able to maximize the diversity of these clusters with respect to whatever variable you're talking about. So parameter choices, the many ways to use harmony. So yeah, you can use harmony. The way that I use it is usually harmony matrix. I just pass a PCA matrix, a metadata table, and I tell it which variable to regress out. You can also use it with Seurat. This is how most people use it. You pass a Seurat object, and then in the metadata table of the Seurat object, there will be hopefully this batch column, and you say, okay, remove this batch column. You can also use this in ScanP in a very similar way. This has also been applied to medical records data. It's been applied to spatial transcriptomics and to ATAC, and all of those packages have their own usage implementation of harmony. Okay, so connecting the model, right? So this is the regression model that we've been talking about, right? So in this toy example here, we have these different arrows, right? And these arrows are just linear correction factors, linear factors really, right? So what we're trying to do in harmony before we correct for anything is learn this regression model, where we try to explain y, which is the location of the cells in PCA space, so Y is the PC components, as a function of intercept, which is some kind of batch-free thing. And then for every batch, we can try to learn its batch correction factor. And because every cell can belong probabilistically to multiple clusters, we want to take a weighted average over all of the possible batches, over all the possible clusters that that cell can belong to. And so this is the full regression model. So how does this connect to when you're running this in Seurat, for example? Right. So in Seurat, when you pass a Seurat object, you've already done PCA. So Seurat object has this PCA, and that's the Y component here. Group by vars in run harmony, you're defining which variables to learn beta for. You could learn for batch, you could learn for donor, you could learn for species, right? And you'll get, and that sets up this formula. And because this is a regression, this is a great reminder. As far as I know, harmony is the only integration method that's able to do this, that's able to simultaneously model and regress out multiple batch variables. So if you have batch and donor, let's say you multiplexed, right, then you can remove both of them. And this is what happens to the equation. You just add a separate set of betas. Number of clusters, you can also set in harmony, and that just sets the number of clusters. So the number of beta terms, number of local neighborhoods. Cool. So let's walk through 3 pretty quick use cases. I think we'll actually have a lot of time for questions because they're pretty simple ones. So I think this is the use case that everyone has used or seen, right? It's just I have 10 patients. I profiled each one with 10x in its own separate lane, and now they all group separately in my UMAP, right? So this is real data from from one of our early studies, and this is horrible, right? This tells us that there's no reproducibility across donors unless we integrate. And so for this case, simple. We just tell Seurat what the variable it is we want to take out. We take it out after we run PCA, before we run UMAP, before we run clustering, and now it's corrected. Now I'll talk about 2 maybe less common use cases. One use case, and this is kind of something that we're considering writing a perspective paper on because it seems to be a very pervasive problem, that I'm trying to integrate 2 data sets, but they have different gene names, right? What do I do with that, right? I have several solutions, and the solutions really depend on why you have different gene names. The first reason that you have different gene names is I downloaded 2 matrices from Geo, and I want to put them together. And I would say that in this case, this is kind of a dangerous case, right? Because this could be very often the reason that they have different gene names is because they were aligned to different genomes entirely, right? In which case I've introduced unnecessary batch effect. Or they were aligned to different genome, they were aligned to the same genome, but they were annotated with different GTF files, right? So I was surprised to find when I dug really deep into this is that, you can align two things to GRCH38, but there's something like 20 ensemble annotation files. And and the number of genes that are different between those can be thousands of genes that are different between those GTF files. And so that introduces unnecessary technical noise into your integration problem. And in that case, the best thing to do, if it's possible, is to get the FASTQs or the BAM files and remap them to the same genome version, requantify them with the same GTF files. Because that's the thing that's going to get you the most accurate results when that's possible. Or you can use this tool that we also developed, which I think answers one of the questions in the queue, for reference mapping, right? So we found that reference mapping is a bit more robust to this problem than de novo integration. But that's a separate conversation. Scenario 2 is I have 2 matrices, they have different gene names, but I can't avoid it, right? So one scenario that comes up is I have 1 matrix is of human genes and 1 is of mouse genes. There's no way that I can get them to have exactly the same genes because they have to be mapped to separate genomes. So in this case, then I would find, the best possible way to map them through orthologs. humanize the mouse genes, or turn the human genes into mouse gene, or or either way, and then treat them, and then concatenate the data sets, treat species as a batch variable, and then integrate over species. Or again, use reference mapping with our Symphony tool. The third case, which is one that I think it's one of the more interesting ones and that we've used, and this is actually from a paper that we just got accepted last week into this new journal that probably no one's ever heard of, called Med. It's the Translational Medicine Journal from Cell. And in this case, our situation was we sequenced a lot of things. We didn't just sequence 10 donors with the same disease from the same cohort with the same technology. We sequenced almost 80 donors from 4 different organ systems. Some were FACS sorted, some were not FACS sorted, from 8 different conditions, some paired samples, some non-paired samples. So just lots and lots of complexity, more than just one batch effect. And so when we do all of this, when we just do a naive PCA map, we'll see that the cells group by tissue first, and that's the color on the left side of panel B. And then panel C says that I colored every donor by a different hue of yellow, different shade of green, different shade of blue, different shade of red. And we can see that even within the tissues, the cells group primarily by donor rather than by cell. Right, and so in this case, what we did was we ran Harmony and we just integrated over both things. And in this case, we really needed to be careful about downstream analyses, but I think that's outside of the scope of this talk. But Harmony was able to handle both effects, right? It integrated the tissues, got them into the same space. And if we looked within each tissue and we looked at the clusters within each tissue, there was really nice representation across donors within every cluster. So we successfully integrated both donor and tissue in this case. So that's actually where I'm going to leave it. I think we have a lot to discuss. I'll leave it with a plug that we're actively hiring computational folks who are interested in analyzing data from inflammatory diseases. We have lots of spatial data coming in. We have a side project on DNA methylation and aging. And of course, we're open to working on pure integration algorithms and supporting and building on Harmony. So yeah, if you're interested, please check out the website and contact me.
Thank you so much, Ilya. Are we coming to the QA section now?
Yeah. So now we can see where we left off.
The first question I'm trying to get the space before it run out is when you run with donor and tissue at the same time, you sequence everything. Is donor plus tissue, is it like you do batch effect, try one at a time, or this is the multiplications of donors and tissue?
Both. Yeah, we modeled both at the same time because they're not independent variables. In this case, they're actually nested variables because we only had one donor for every single tissue, for every sample. We didn't have paired tissues for any one donor. So in this case, yeah, this is the way that Harmony handles this is with this this kind of regression model where we simultaneously model the effect of both. And then in this case, we chose to take out the effect of both, to remove the effect of both. But you could actually choose to keep one and remove the other if that's relevant for your downstream questions.
Right. Let me read the questions out. So
all right.
One question is from an anonymous attendee. Is there any other way to access how successful my Harmony integration was, except for the plotting?
Oh yeah. So that's a good way. So it's successful. So I guess there's 2 broad metrics for success. One metric is I've successfully mixed my cells, right, locally. And so there, plotting is a great way if you have a small number of batches. If you have, 3 to 5 batches, you can give each one a color and you can see if they're well mixed. And it's actually a good way to, it is a good first pass, because a lot of times, what will happen is, or what you really want at the end when you do a good harmony integration is this, where you have really locally well mixed things, right? Sometimes what you'll get is that things are sort of close to each other. 2 batches, get sort of grouped together, but they're not like super well mixed locally. And that's a sign that something went wrong.
Okay.
And so that's a good way. If I have 100 data sets, that's not going to work, right? And so I can't just color by 100 colors. So there's many metrics. We developed the Lizzie metric in the paper. Lizzie metric is really nice because it tells you for any cell, how many data sets are well represented in its neighborhood, right? And so that's another way to tell, if you have too many. And yeah, data sets or downstream differentially expressed genes, you need to be careful about how you do differential expression at the end, right? Because you can't, after you integrated in your differential expression model, you can't just ignore the effective data set anymore. You have to model the effective data set within cluster as well, right? And so I think that's what I would say is be careful how you do these models. You can't, so a lot of times you can't just do naive Wilcoxon or t-test because it doesn't take into effect these donor effects, batch effects. Okay.
Right.
Next question.
The question also is, when we deal with small cell types, they have very small cluster. What is the minimum size per cluster that you consider?
I think that's really not something that harmony will. I'm going to rephrase this. Harmony is not your bottleneck. Your bottleneck is what you do before harmony, right?
Yes.
Because if you do your, you do your normalization, you do your scaling, you do your dimensionality reduction, and if that small cell type is distinguishable in that low dimensional space, right? Harmony is not going to do anything to remove that cell type, right? So it's really everything before harmony that I would pay attention to.
Thanks, Ilya. The next question is that, is there any assumption on non-linearity in the model at some point, even though it says multiple linear model, but it seems that there is more to just linear regression, some sort of topological modeling. That's one.
Yeah. I mean, this was, like philosophically one of our interesting points, right? Like, I assume that when you mean topological modeling, you're saying that, these data lie on a nonlinear manifold, right? And so, you map T-SNE manifold alignment, local linear alignment, all of these algorithms that are designed for manifolds rather than things that lie in Euclidean space. The philosophical motivation for this is that manifolds are locally linear, right? But globally nonlinear. And so if, in theory, every manifold should be able to be modeled by Gaussian mixture models because they're locally linear. And so that's philosophically why we think, or why we thought of this approach, right? Is because if manifolds are locally linear, let's model the local linear relationships. That was it.
Yes, there's another question that I don't understand so much. So I predict, I just read it. Is it possible to predict if I need to correct for batch effect in case I don't have the cluster assignment?
Yeah, I mean, usually we don't have cluster assignments before we do batch effect correction. We'll do this kind of plot where we'll see, this, right? So here, I know which cell types they are because I've seen this plot a million times, but we, at this point in the analysis, we don't know what the clusters are, right?
Yes. Yes.
Yeah, and so this is before clustering.
There's another question from Volkan Servin.
Okay.
Would harmony work on cell types that do not really cluster or separate it well?
Yeah.
Yeah, for example, bone marrow cell type don't cluster well.
Yeah. Yeah, so I can say that we've definitely used things, harmony on things that, bone marrow. You have a, you're modeling, hematopoiesis, which is a very continuous process, rather than analyzing terminally differentiated cells. So of course they're not going to separate well because they actually live on a developmental trajectory. So in the paper, we did actually analyze a developmental hematopoietic trajectory in the manuscript, and I wish I put it in the slides. I didn't, but I can point to this in the toy example. I can point to clusters 3 and 4 here. We can imagine that this is some kind of trajectory that goes from cluster 3 to cluster 4, and the way that we handle this is by using soft clustering, right? And so we don't force this trajectory into discrete clusters. We keep the trajectory shape.
Well, one particular example, usually CD4 and CD8, I don't see them separate so well.
Yeah.
All right. So another question. Can you comment from Matthew Bernstein? Can you comment on what task would use the harmony transformed expression value versus the raw normalized expression value?
So, well, first I'll to avoid any confusion outside, we right now don't have a method to do harmony transformed expression values, right? So right now harmony only works, or we only recommend using harmony on low dimensional embeddings. We have a few things where it would be useful. So we can imagine RNA velocity is probably somewhere where you would probably want to correct, because the actual gene values, not just the PC embeddings, because you learn the model on the gene expression values. Clustering, UMAP, TSNE, DDR tree for monocle, they can all be done with PCA embeddings. They don't need corrected gene expression values, right? But there are some algorithms that do.
Yes. So the another related questions from myself is that, would all these Bash effect removal, the value, the transform value after Bash effect removal, what are they useful for except visualizations and grouping cell type together?
Yeah, so once we've corrected the PCs, everything. So visualization, you've mentioned, right? UMAP and TSNE. Grouping cell types together, clustering, is done on the harmonized PCs. You can do trajectories with popular algorithms. So Slingshot, DDR tree, they can all work on PCs, right? Because they just need a KNN graph as input, basically. And so all of these tasks can be done with that. Oh, and there's actually a family of algorithms that we didn't really even touch on, but that do case control analysis without clustering, right? So I'll put a plugin for this CNA algorithm, covariant neighborhood analysis. I think that's one that's where harmony has been very useful for us. Also, is applying this one. So I would check that out as another use case for after integration.
Thanks, Ilya. The next question, I think, from a wet lab biologist, from Elisa Rosati, if I want to correct for multiple Bash effects, partially correlate, is it a problem? For example, I have 6 samples prepared in 3 different days. 4 of them were sequenced together and 2 not. What is the best correction strategy? Correct for both sequencing run and experimental day?
I mean, the first thing I would do is plot it, right? Plot it and see how they separate. We found that sequencing batches don't really create batch effects. in our data sets. Running things in different 10x lanes, running things on different days. I guess it's running things in different 10x lanes. That creates batch effect. We found that sequencing in different lanes is not as much, right? But anyway, but as a general answer to your question, plot it, color it by your variables, and it'll become obvious to you what the biggest source of batch effect is.
Yes. So visualization is important here.
Very, very, yeah. No, it's always, right? Yeah, you can't rely on statistics alone.
Yes, that's right.
Yeah.
The next question: if donor is used for harmony, will that also address the dataset batch?
Depends on how donor is related to dataset. If donor is dataset, then yes. If donor is nested within dataset, then maybe you need to do both, or maybe you choose not to. When you multiplex donors into the same 10x lane, you drastically reduce batch effect between the donors, right? And so a lot of times we just don't actually correct for donors if they're all processed in the same, multiplexed into the same 10x lane. Sometimes we leave them as is. Sometimes we integrate them anyway.
And the next question from Nikolai. As far as I understood, to start the first iteration of linear batch correction, you need to know the nearest neighbor across batches that belong to the same cell population or cluster. What if the cells look like nearest neighbors because of the batch? In other words, if the gene expression variation due to the batch is not orthogonal to the biological variation, but collinear, how would harmony or even MNN solve this?
Sure. So I'll say that one thing that's very different from a lot of the other, especially early batch correction algorithms, is that we don't use KNN graphs at all, right? So I would say that Seurat, CCA, MNN correct, Scanorama, BBKNN, they're all kind of in their own family of batch correction and integration algorithms, because they do basically what Nikolai, you're saying, is they find these nearest neighbors, right? And in fact, a lot of them have kind of converged to the same algorithm in a sense, right? Because they all use mutual nearest neighbors, they all use the same heuristics for data set ordering and everything. Harmony does not use nearest neighbors at all, right? It uses Gaussian mixture models instead of nearest neighbors. And so I think that's what makes it super fast compared to KNN things. The second part to the question is orthogonality, right? So we don't assume that. And it's actually, sometimes it happens. Sometimes it is really orthogonal. PC1 is cell type, PC2 is batch effects. Most of the time it's not. Most of the time there's batch effects that are very much correlated with cell type. In fact, that's the interesting cases. And Harmony seems to do fine there. We haven't seen the case yet where batch effect makes a T cell look like a B cell from another batch. As an extreme example, we haven't seen anything like that yet. But we definitely don't assume any orthogonality.
Right. And the next question from Dylan Richards. Is harmony suited for addressing batch effect from the set with different cells?
Very much. Yeah, very much. We've done exhaustive negative control examples also, where we take one batch that has all one cell type and another batch that has all a different cell type, and we make sure that harmony does not match any of the cells across the batches.
Yeah,
right?
I think this question was also coming from the background of the mutual nearest neighbor, assuming that two batches need to have a shared thing so that.
But we don't assume that at all. No. They can have 0 shared, and then we won't integrate anything at all. We just won't touch it at all if they don't have anything shared. We've made sure that works a lot of.
This is good to know as well.
Yeah. And so.
Next question. Can you give high-level explanation the difference between harmony and SC transform in Seurat?
Yeah. SC transform is not really a data integration method. It's a normalization and scaling method, right? So SC transform is a how do I say this? It's a generalized linear model. That's the regression, right? But it does not account for nonlinearity due to manifolds, right? So it does not, it assumes that genes have one peak, basically, right? It doesn't use mixture modeling, it doesn't use manifold alignment. So I would say SC transform is a great normalization method, but it is not a data set integration method.
Yes. And the next question from Jonah. Do you see problems integrating different data sets generating from different technologies? For example, single cell nucleus was single cell sequencing, or even take one step further, whether single cell RNA sequencing was another model of spatial sequencing.
I think there's tons of problems and everything, but the benefits outweigh the problems so much, right? That is definitely worth doing, right? Especially for spatial and single nucleus and single cells. So, right, single nuclear a lot of times for some reason doesn't capture immune cells very well, and single cell obviously doesn't capture weirdly shaped cells like muscles and fibroblasts very well. And so it's a great design strategy to use single nuclear and single cell and then combine them based on their shared cell types. I think it's,
Yes. What is the main difference between Seurat integrate function and harmony?
So this is something I mentioned before, right? So Seurat, it finds anchors. Seurat essentially can integrate two data sets at a time, right? And then it adds in a third, adds in a fourth, adds in a fifth to do multiple. And Seurat uses anchor base, so it finds cells that it thinks are very similar across two data sets and then anchors them together. We don't use anchors, we use regression and mixture modeling.
So in the previous one, it actually depends on the order that you integrate?
It does, yeah. That was the big result from the Scanorama paper in Nature Biotechnology a few years ago, was that the order really matters. And then everyone fixed that problem, to have a better order. But that's the thing. So yeah, you can imagine in the worst case scenario, if you try to find anchors between 1 million cells and 1 million cells, you're doing 1 million or 1 million squared comparisons, right? But we never compare one cell to another cell. We only compare cells to a centroid, right? And so that's what lets us scale much more efficiently to big data sets.
Also, next question. Can you run harmony with two very different data sets? Do you need to downsample them?
Yes, and you're right to be worried. I would say that if you run harmony with one data set that's just vastly bigger, let me actually augment the question. Harmony itself should be fine. I would worry about the steps leading up to harmony, right? So if you do gene scaling, PCA, those steps, if you don't account for different sizes in the data sets, they're going to be very dangerous. If you're interested, I would read the paper. It's actually on bioRxiv and it'll be in Med soon, but the paper that we did with the multiple tissues, we had 70,000 fibroblasts from synovium and 1,000 from lung. So we really had to deal with this issue. We ended up using weighted PCA, and that solved like 99% of our problems. So yeah, definitely very important. You can downsample. I don't like downsampling because we're throwing away data, but you can use weighting strategies to give both data sets equal weight.
Right. Do you have any thoughts on handling tumor microenvironment studies, the true donor-Bush effect within immune cell type, but the malignant epithelial cells are not accurately cell type?
Yeah, I mean, cancer is weird in that way, right? Because no two cancer cells should be alike across different donors, right? But at the same time, if every tumor was 100% different among patients, there would be no therapies that would translate across patients, right? So yes, they're different cells, but they probably have very similar underlying processes, right? So maybe to go back to Bob Weinberg.
Yes,
I hope so. And the hallmarks of cancer, right? You can see this population.
We run out of time.
Okay.
Exactly 1 hour. And this is an amazing talk that I was just trying to speed up answering the questions, but asking the question, but I couldn't speed up it because I have to listen to the content because this is so informative talk.
Oh, great. No, thank you for the opportunity. And thank you everyone for the questions.
And the rest of the questions we will send to Ilya, and he can answer it, and we can distribute it via emails. But again, thank you so much, Ilya, for this very, very informative talk. I myself learned a lot from this, and you know, the task of reading the questions and actually distract me a lot from hearing what you're saying. And you know, arguably, you know, there are a lot more things to do with partial effect, and I think you know, the field is just moving so fast. And and I would personally like to thank Ilya for such a huge contribution for the field.
All right.
All right. Thank you
so very much. Yep. Look forward to answering everyone's questions.
Thank you.
Bye, everyone.
Have a good day. Bye-bye.